have you ever wondered how scientists analyze the structure of proteins? It all begins with understanding the backbone torsion angles, the phi I and psi angles that influence the overall protein structure. These angles describe the rotation of peptide bonds, forming the basis for the fascinating world of protein structures. The phi angle, for instance, is the angle around the bond between the nitrogen atom of one amino acid and the alpha carbon of the preceding amino acid. On the other hand, the psi angle is the angle around the bond between the alpha carbon of one amino acid and the carbonyl carbon of the preceding amino acid. Enter the Ramachandran plot, a graphical representation named after Indian biophysicist G.N. Ramachandran. This plot, created in the 1960s, is a scatter plot depicting the distribution of these phi and psi angles for amino acid residues in a protein structure. This plot has regions known as allowed and disallowed. Allowed regions are where the majority of protein structures fall. They represent combinations of phi and psi angles that are sterically allowed and energetically favorable for stable protein structures. In contrast, disallowed regions are where certain combinations of phi and psi angles are sterically unfavorable. Proteins with backbone torsion angles in these regions may experience clashes or strained conformations. The Ramachandran plot is more than just a scatter plot. It's a tool that helps researchers and structural biologists assess the quality of a protein structure. If the phi and psi angles of a protein structure fall within the allowed regions, the structure is likely to be more stable and accurate. Conversely, outliers in the disallowed regions may indicate errors in the protein structure or inaccuracies in the experimental data. Beyond analyzing protein structures, the Ramachandran plot also aids in refining protein models. Researchers use these plots to validate and refine protein structure models generated through techniques like X-ray crystallography or nuclear magnetic resonance. This structural refinement involves adjusting the protein backbone angles to align them with the preferred regions on the Ramachandran plot. In essence, the Ramachandran plot is an invaluable tool in the world of biochemistry and structural biology. It provides a snapshot of the stereochemical constraints that govern protein conformation, aids in refining protein models, and offers insights into the quality of protein structures through the analysis of backbone torsion angles. So the next time you look at a protein structure, remember the humble Ramachandran plot and the pivotal role it plays.